sulfate polyacrylamide gel electrophoresis. So basically this is used as a biochemical technique in the lab to separate macromolecules. So basically DNA, RNA, proteins, and they separate based on size and charge. The proteins move toward the electrode of the opposite charge, hence how they separate. And the rate of the movement that occurs is governed by the relationship between physical characteristics of the system and the proteins. So these are the affecting factors. It can be electric field, pH, temperature, the ion type of concentration and charge of the proteins, anything like that will affect how gel electrophoresis will run. So today in our lab we will be examining the vegetable protein ferredoxin NADP plus reductase. And this in vegetable or most plant-based organisms, it catalyzes the following chemical reaction, which is here, it's also in protocol. And this reaction occurs as a process of photosynthesis, in the process of photosynthesis. And this is just our structure, beta helix structure of the reductase. So, for electrophoresis, we will need separating and stacking gels. I'm not exactly going to cover that because we already have ordered the ones that are already prepared. So basically the steps for running SCS page gels. We have the gel cassette where we put our gel into the chamber and the tank. Now we'll place our gels, we'll only run one, so one will be placed on one side, closing the casket and placing it into the chamber, and that will also go in the tank. Where we have two buffers, anode and cathode. So the cathode buffer will go in between the cassette of where the gels are. The other buffer will go on the outside in the tank, filling it. Pretty much there's a stop mark in most of them to fill. From there, we will inject our samples. And from injecting our samples, we will connect our tape to the power source, run about 100 volts, about 30 minutes, and our protein should separate. I also have a video to help guide. It's kind of short. There's not a lot of. Here's my sound. We can just get it. So there's the cassettes in the tank and his samples. So then he's going to assemble. And that is where your gel will already have those pre-made. Put that in. It's in there tight, like so. Since we're only running on gel, I'm going to kind of put another gel should go kind of like a plastic. This way. Alright. Let's get the steps, so we're putting it in.
What is this?
Into the tank. And now we're adding the buffer into the inner chamber. And once there, we'll add our samples.
CHEM?